Hello everyone, welcome back to my channel Indu Biotech. Hope you all are well. In the last video, we have discussed about the theoretical part of the BLAST search tool, and today we will discuss about the practical of the BLAST tool. So let's we start. You can go to the BLAST section through the NCBI home page. Just I type the NCBI and NCBI home page by clicking on the BLAST, right? Or you can go through the searching web and simply you type the BLAST, 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 and we just by clicking on the tab we get the home page of the BLAST. This is the home page of the BLAST tool, and home page, or you can say this is the main page of the BLAST search tool. Here different search is available, like the nucleotide blast. In this search tool your query will be the nucleotide and results also will be get in the form of nucleotide. And this is the protein blast search. Here your query will be in the form of protein sequence, and your result also you will get the result in the form of protein sequence. And this nucleotide blast and protein blast is generally used in the BSc or MSc level, and blast X and blast T blast, and it can be used either in the MSc or BSc level, or mostly used in the research. This is high level of the similarity search tool. In the blast X, the your query will be in the form of translated nucleotide, and result is in the form of protein sequence. And in case of the T blast M, the query will be the protein sequence format, and result you will get the result in the form of translated nucleotide, right? In the below you can see the different specialized searches is available. For the example, it is the global align, primer blast, smart blast. So I will discuss all of these in later. Now we move to the home page, main home page. Right, when you click on the nucleotide blast, you will get the main home page of the query page of the blast tool. And here is I am selecting here on the top, and there is a different search is available: blast N, blast P, blast X, T blast and T blast X. So here I am using the gene ID. Here is the gene ID 4363, I think 4363, right? Here 4363. So you can here add in this inter query sequences section, you can enter your query sequence in the FASTA format, or accession number or gene IDs you can enter in the query sequence section. And you the another option is also you can upload in the file format. And in next it is the query query subrange option. Here you can limit you can, by this section you can limit your sequence search. For example, if you want to enter 1 to 500, 200, 300 something, here I am adding the 500. That means 500 500, it means it limit the sequencers to the first 500 nucleotides of your DNA or first 500 of amino acid of your proteins, right? So you can, and in this you can write the name of your search title, as it is automatically appear. And this is the choose search set. In the choose search set, it is the database. Different database is available. The standard database are RNA RTS database, genomic plus transcript database, beta coronavirus database. And by clicking in the drop down section, there is the many option is available. You can choose as your requirement. And this is the question mark section. In this section you can check the what sequences is available in the database. And this is the organism option. This is the optional for you. And if you want to add, you can add, like the I am adding is a human chromasis cerevisiae. Chromasis cerevisiae I am adding. Or if you want to add additional organism, you can add definitely. And this is the interest query option. This is also a optional. This is also a optional. You can enter the interest query syntax to limit the macromolecule type of the searches. And the last one is the program selection. If you want to identify for the nucleotide sequence, then you should go through the highly similar sequence mega blast, right? And because the mega blast is a specialized designed to the find long alignments between the various databases sequences. So if you want to go for the protein search, you should go through the BLAST P. You should go through the BLAST P program, because this is the standard BLAST P, and identify a query amino acid sequence and find the similarity sequence in the protein database, right? And this is the another important parameter. You can also choose this parameter. If you in the max target sequences. Max target sequences. That means this program you will, so in this program you will see the maximum 50 to 100. If you set 100, you will see the result maximum 50 of the align sequence in the result page, right? Now if you click on the this show result in a new window, you will get the results in new window, right? After that you should click on the blast. Here I am changing, this is the blast pin, but this id I have given is the nucleotide gene id. So I am going to the blast and just will click on the blast, right? After clicking on the blast, it it will take some time to process your query. Yes, once the search is complete, new window page is appear with your final results. So this is the results of blast. That's all guys for today's. Thank you so much, and result we'll discuss in next video.